So the good news is that your exam 2 results are back, so you can look up and you'll see, and you can check your grade. It wasn't too bad, and our TA will have your exams for the pickup, hopefully by the end of the lecture. And of course, I know that previous instructors already emphasized that. This class is graded on the curve, so do not be afraid of those points that you're having right now, because you can always improve. There are 2 more exams, so there's lots of room for improvement. And yeah, like I said, it's graded on the curve, so do not worry too much. You have a question? The breakout sessions. Yes, the thing is that I have to consult with previous instructors how they did it. I'm not sure if they allow us to upload those information or the breakout. They did it? Alright, so we have to make it consistent. So let me check with previous instructors, and if they allow, I can post those and the ELC, but I know that in previous years we were not allowed because they are using the same material year after year. Another question. Say it again? I'm sorry, maybe I was saying... Okay. I was saying about second exam, so the results for the second exam are back. The second one, from Dr. Shen. Alright, today we're gonna finish talking about chromatin remodeling. There are a couple of slides that I want to go over, and then we'll move on to stem cells and cell reprogramming. So in the previous lecture, I talked a lot about chromatin structure and chromatin remodeling, various mechanisms for chromatin remodeling, and that chromatin in our cells can exist in 2 distinct conformations. We can have more condensed chromatin fraction and more open chromatin fraction. And those states in cells are tightly regulated, and there are special groups of enzymes that regulate transition between condensed chromatin and more open chromatin. And I told you that there are histone acetyltransferases, HATs, which specifically put acetyl groups on N-terminal tails of histone. And this process allows for opening of chromatin, and this can allow for gene transcription. And another group of enzymes, histone deacetylases, HDACs, which can kind of reverse this process and can convert open chromatin back to the condensed state by removing those groups. And I wanted to kind of go deeper into the mechanism, how it happens. And this figure is to illustrate you this opening and closing of the chromatin in the context of the gene transcription. And in this case, we have a gene. So this is the promoter region for the gene called nitric oxide synthetase. And this gene is differentially expressed depending on the cell type. And it's transcriptionally silent in the smooth muscle cells. And this is the promoter region and the chromatin conformation of the promoter region for this gene in the smooth muscle cells on the panel B. So let's focus on this. So in this, DNA is tightly wrapped around the histone octamers, and those nucleosomes are packaged very closely one next to another. And what else we can see here? We can see this enzyme in purple, HDAC, sitting here, which would remove all of the acetyl groups and would promote condensation of the chromatin in this region. We also can see a DNA methylation. DNA methylation is another mark that signals the cell to condense or silence the chromatin. And in this context, this DNA is not open, is not accessible. and pretty much the gene, the promoter region for this gene is silent, so there is no gene expression. And here on the panel A, what's happening, under special circumstances in those different cells, vascular endothelial cells, there are some signals coming in, and they are telling the chromatin here to open up and to change the conformation to allow for the gene expression. And what's happening, one of the first events that can happen here is the recruitment of histone acetyltransferases, HATs, and they're gonna come here to this region and they're gonna bind to the octamers, and they will put acetyl groups. So acetyl groups carry negative charge, and they can interact with the negatively charged DNA, and this will cause a repulsion. So they will start kind of like removing or relaxing the DNA that is tightly wrapped around the histone octamer. So this region will start like slowly opening up, and we know also that acetylated histone pairs are the signal or marks for recruitment of ATP-dependent chromatin remodelers. So those big chromatin remodelers can be recruited to this region, and they can further facilitate opening of the chromatin structure, loosening this region and exposing important regulatory elements. So here this DNA is much more accessible, and RNA polymerase can bind, recognize its binding sequence, and all of those processes can allow for efficient gene transcription. So acetylation is very important in regulating gene expression. And in this context, acetylation imbalance can influence transformation of normal cells to cancer cells. You may know that in our healthy cells, there are two main categories of genes that protect our cells from cancer formation, and those are called tumor suppressor genes and proto-oncogenes. And both categories of genes have to be expressed in normal cells at specific levels. And what's happening when acetylation is kind of like off balance, we can have downregulation of tumor suppressor genes, which would kind of could initiate tumor formation and progression. And on the other hand, if we have a hyperacetylation of specific region, and hyperacetylation happens in the proto-oncogenes, those events can also initiate tumor formation and progression. So acetylation in chromatin and the balance is the key to maintain cells happy and healthy and prevent cancer formation. And this is another very interesting phenomenon regarding the chromatin when we talk about heterochromatin and euchromatin, and in the context of gene expression. So some genes can change the position in the nucleus depending on their expression status. So in this figure, we have an example of a gene, and there are two copies of this gene. So in this top panel, the gene is silenced, it's not expressed, and it's colocalized with the heterochromatin or silenced fraction of the chromatin, which we said that is very abundant and on the nucleus periphery. So here, in this situation, this gene is silenced. But when the chromatin changes in the promoter of this gene, it can allow for active expression of these genes, and what's going to happen, those two copies will change location, will change position within the nucleus. So now we can see them, they kind of are transfer more to the nucleus center, where we have a more accessible, more open chromatin. And this environment now will facilitate efficient gene expression. And our understanding of the chromatin structure is increasing, and in the recent years we have more and more much better idea how chromatin is organized in our cells, and we also have the ability to look and model the chromatin in three-dimensional way. And so this is a very nice model, and you have this figure in your textbook. So if you need to have more information about that, I encourage you to look up in your textbook. But this model shows you the chromosome. So this gray long thing is entire single human chromosome. And here it shows you how this chromosome is organized in the interface chromatin. So interface chromatin is the chromatin in probably majority of our cells, the cells that are not currently dividing. So those chromosomes are slightly decondensed, but not totally, not completely decondensed. So I think I said in the previous lecture that chromosomes, they have a occupy specific space within the nucleus. They are located within the chromosome territory. And this is a very nice model showing how this entire chromosome is organized. So it has a specific place within the nucleus. And what you can see a specific very nice pattern of those, and those are called a fractal globules. So the entire chromosome is kind of decondensed, and it forms those fractal globules. So here in this rainbow color, you can see there is a region of 5 megabase, and this represents a single fractal globule. So this is a really nice pattern, and it kind of reminds me the beats on the string, even though this is a completely different scale, because here we have an entire chromosome. But when we look at this pattern, it's very regular. So those fractal globules are very highly organized, and they are regular. So there is a really nice pattern. And of course, we have lots of tools to study the chromatin, and one of the methods, one of the approaches is chromatin immunoprecipitation, called CHIP, and it could be coupled with high throughput DNA sequencing, and using this method we can map individual nucleosomes, and we can create the maps of nucleosomes in a genome-wide scale. And those maps were created for lower eukaryotic organisms such as yeast cells, simple yeast cells, and also we have those maps for higher eukaryotes like human cells. And this is a brief overview of this approach, what we can do to map those nucleosomes on the genome-wide scale. So first of all, we have to kind of cross-link the DNA and histone octamer, so we can freeze those nucleosomes in place, and then we can purify the chromatin from the cells, and we can shear it, break it into smaller pieces like this, and then we can use antibodies specific for the histone proteins, and we can pull down this fraction. Then we'll purify the DNA from those shorter regions, so remove the histones completely, purify the DNA, process the DNA ends. We can attach some specific adapters to the ends of this DNA, and then we can subject this DNA to a next generation sequencing, which is a high throughput sequencing. And what's going to happen, the end point of this process is that we'll get lots of reads looking like this, and we are comparing those reads, comparing those reads to the reference genome. And this way we can map the location of the nucleosomes on the genome-wide scale. So this is one of the approaches. It's very commonly used, and this is a very good way to understand where the nucleosomes are, understand more about their occupancy and their dynamic properties. And this is the final slide for the chromatin, kind of like a summary. And I really like this slide because it shows a very important information. So on top, we have just the naked DNA without any proteins. So what it does, it's just the storage of the information, the storage of the genetic information. And then when we have the chromatin, this information is organized. And when the information is organized, this have a meaning. This can respond to different signals. It can respond dynamically to like surrounding environment, to changing environment, because DNA on its own is not able to do much. So we need all of those proteins. We need to organize this information. And on top of this organization, we have another layer of information in the form of those histone tail modifications. So it looks like there is a code. On top of the code, we have histone, we have DNA code first, then we can talk about the nucleosome code, then we can talk about the histone code. So it looks like there is a code on top of the code on top of the code. So epigenomes, they have so many different layers of information, and this is all too necessary to regulate different processes within the cells and dynamically respond to changing environments. So this is all necessary. Are there any questions or comments or ideas? Okay. So now we'll move on to stem cells. So stem cells, what are stem cells? All of the cells in our body, we call them somatic cells, so our skin and all of the cells that we have in our body are somatic cells. So all of the somatic cells were derived from the stem cells. And they were derived from the embryonic stem cells. So this whole panel shows the top portion of this panel shows a very early embryonic development, and you don't have to go into the details, of course, this is not the developmental biology, but I wanted to kind of emphasize here the source of the embryonic stem cells and where they are. So they are found only at this stage of blastocyst, which is very early embryonic development. This is the 6 days post-fertilization, and this inner mass of cells are embryonic stem cells. And all of the cells in our body were derived from this mass. So for the rest of the lecture, we're going to talk about stem cell biology. We're going to introduce some important terms, and we'll also talk about applications and stem cell research, and also the regenerative medicine, and how important they are in medicine and in biology, in treating diseases. So there are a variety of different applications of stem cells. And of course, learning objectives. There is a very nice chapter in your textbook, chapter 22nd, and I encourage you to go there. It's not very long, but it's very, very interesting. So all of this information you should find in your textbook. So stem cells. There are 3 major sources: embryonic stem cells, adult stem cells, and induced pluripotent stem cells. And we'll talk about the applications in regenerative medicine, drug delivery, cancer treatment, and human development. So what are stem cells? So we have about 200 different kinds of specialized cells, such as muscle cells, nerve cells, bone, or skin cells. And I already said that all of those cells came from embryonic stem cells. So a stem cell, by the definition, is a cell that is not specialized or not differentiated. So it's the precursor for all of the specialized cells. And this last part, the stem cell can differentiate to specific cells, and the differentiation is tightly regulated process, and we'll talk about that in a little bit. But first, in order to understand the stem cell biology, we have to kind of understand what are the unique features of stem cells by comparing them to the somatic cells. So how stem cells are different from somatic cells in our body? And these are like 4 key unique features, and you have to absolutely know those 4 key unique features for the exam. So first of all, the stem cell, like shown here, is this cell that is unspecialized. So it's more like a precursor, or it doesn't have a specific function. And it can go, it can go and it can self-renew. So it can go and divide and make more of the stem cells, which are unspecialized. So it can keep this unspecialized state. So it can proliferate and make more stem cells, or it can choose a different path. It can choose a path and it can differentiate to specific cells. So first of all, stem cells are different from the somatic cells because they are unspecialized. And second, they can differentiate to different cells. They can self-renew. Our somatic cells, they don't have this ability for self-renewal. And very important feature of stem cells, they can proliferate indefinitely, so forever. None of our somatic cells can do that. So they are pretty unique in their characteristics. And I've mentioned there are 3 different types of stem cells, embryonic, which are also known as pluripotent, because they can form almost any cell type in the human body. We have tissue-specific stem cells, which are called adult stem cells, and they are multipotent. And make sure you understand and can distinguish those 2 terms, pluripotent and multipotent. So multipotent can form only specific types of cells, such as blood, brain, liver, and they have only limited ability to create different cells. Pluripotent can form any type of the cell. And of course, the 3rd category is very interesting. Those are induced pluripotent. They are engineered by scientists, and they can act like embryonic stem cells. So they are created by taking the adult cells and reverting them back to this state, to the pluripotent state. And we'll talk about the process of creation of induced pluripotent stem cells. And so the next question is, what is this? So we are using human embryonic stem cells in research. This research is very limited because, of course, there are lots of ethical concerns and considerations. But yes, there are some sources of human embryonic stem cells which can be used in research. And typically, researchers obtain those cells from the IVF embryos. So those embryos are leftover embryos after the IVF in vitro fertilization procedures at the in vitro clinics. And so this panel shows a very early stages of the embryonic development from day 0 to day 6. So all of those stages happen in the in vitro clinic when, for example, a couple who wants to have a baby, they decide to do in vitro fertilization. They go to the clinic, then the eggs are extracted from the female body and sperm is donated, and then the process of fertilization happens in the petri dish, and then it continues in the petri dish up to day 6. So all of those stages are happening at the in vitro clinic for 6 days. And here at this stage, at the stage of blastocyst, it's day 6, we have here this inner mass having the embryonic stem cells. And at this stage, the in vitro clinic is asking the couple how many of this blastocyst they want to transfer, in order like how many babies they want to have. If they want to have just one baby, they would transfer just one blastocyst and would freeze the excess. And typically, in this process, there is quite a large excess of those embryos, so they will be frozen. If they are not used for the pregnancy, they will be frozen and kept in the liquid nitrogen either forever, or couple can decide and donate them for research. And some people decide and they want to donate those embryos for research, so researchers can work with those cells. But of course, like I said, there are lots of considerations and ethical issues associated with this, because in the process of obtaining those embryonic stem cells, the embryo will be destroyed, like completely destroyed. But like I said, those cells are extremely potent because we can maintain them in the laboratory and we can control them by growing them in specific conditions, and we can differentiate them to pretty much any type of the cell in the body, which is really powerful. And of course, they can be used in to restore the organ function in animal models, and they can be used for variety of different in attempt to treat variety of different diseases. And they of course can be differentiated. And here this picture shows heart cells that were derived from the embryonic stem cells in the culture, in the petri dish, and they were able to create heart cells that were able to so they were like beating heart cells, that they were fully functional heart cells on the petri dish. And in our bodies, we have tissue stem cells. A variety of different tissues have their adult stem cells. So they reside in most tissues of the body, and they are very important for the tissue repair, replacement, and to maintain tissue homeostasis. So we have stem cells in our brain, in bone marrow, in our peripheral blood vessels, skeletal muscle, skin, liver. So all of those tissues have adult tissue stem cells. And those stem cells are very difficult to isolate, and they are not very abundant. So they are very small. There are very small numbers of those cells, but they've been already used to treat patients, especially with the hematological malignancies and also different diseases of the immune system. And yes, again, those tissue stem cells, adult tissue stem cells, are critically important to maintain tissue homeostasis, to remodel our tissues and to keep them healthy and functional. And to give you a perfect example for this, I want to talk about the small intestine and the stem cells in small intestine. So maybe not all of you are aware that the lining of our small intestine is replaced every 3 to 5 days completely. And this is critical to maintain the balance and a healthy digestive system. And this panel here shows the structure of the lumen of the gut. And what you can see here, in green, there is a connective tissue, and on top of it, there is a single layer of epithelial cells. And those are a very specific structure in our small intestine called villi, and they are to increase the absorption area. And when we zoom in here at the bottom, there is a structure called crypt. And what we can see here at the bottom of this crypt, there are stem cells. There are not very many of them, but they are critically important to maintain this entire structure functional. So here at the bottom we have the stem cells, and they divide every 24 hours. And they divide, and they are the progenitors or precursors for all of those cells that are here up. So they divide and they provide those. So here that there are rapidly dividing cells. They divide every 12 hours. And here on top we have a non-dividing differentiated cells. So all of those cells, all of those epithelial cells are derived from the stem cells residing here at the bottom of the crypt. So in our guts right now there is a continuous process and proliferation. All of those cells here, shown here in yellow and blue, they are dividing and kind of moving upwards. And I have a really nice movie. Hopefully you can play it.
The lining of the small intestine, like the lining of most of the gut, is a single layered epithelium. Depending upon location in the gut, the epithelial cells, usually called enterocytes, help absorb nutrients from the lumen of the gut or absorb water from the intestinal contents. In the small intestine, the surface of the lining of the gut is increased enormously by thousands of villi that protrude into the lumen. Here we see a single villus and its internal architecture. The surface of the villus is covered by a single layer of enterocytes, which extend down into the crypt below. Stem cells at the bottom of the crypt, located between Paneth cells, divide and make copies of themselves, and also make transit amplifying cells. The transit amplifying cells proliferate rapidly and move up the walls of the crypt. As the cells migrate upwards, they begin to differentiate into goblet cells and enterocytes. While the cells are moving up the sides of the villus, they carry out the essential functions of the small intestine, notably absorption of nutrients. When the differentiated cells reach the tip of the villus, they undergo apoptosis and are shed into the lumen of the small intestine. The entire process of outmigration and cell death is completed in just 3 to 4 days. The process of outmigration and rapid cell replacement is a defense mechanism against the development of colon cancer, since almost all epithelial cells, including those that have accidentally sustained mutations, are shed within days of their formation. Therefore, the only mutations that can lead to the development of a cancer are those that are retained in the crypt. This dictates that such mutations must block the outmigration of mutant cells from the crypt. The outmigration of transit amplifying cells from the bottom of the crypt depends on a protein called adenomatous polyposis coli, or simply APC. In the absence of functional APC, this continuous outmigration is blocked, leading to the accumulation of transit amplifying cells in the crypt. In this animation of an experiment, APC loss is achieved through an induced gene inactivation, which appears to mimic the mutation that initiates most gastrointestinal tumors. APC is usually required to inactivate the intracellular protein called beta-catenin. The inactivation of beta-catenin permits the differentiation of the transit amplifying cells and their continued outmigration from the base of the crypt. In the absence of APC function, beta-catenin accumulates within the transit amplifying cells, which blocks both their outmigration and their differentiation. The accumulated transit amplified cells do not themselves form a carcinoma. However, they and their descendants can now accumulate additional mutations that will drive such cells progressively to become full-fledged carcinoma cells.
So yes, importance of the stem cells in our small intestine is huge. So we have to have those cells healthy. And regarding the video, do not worry about the physiology. Do not study this video from the physiology level. I won't test your knowledge on the very details and the mechanistics of the gut. Of course, this is not the physiology class. But yes, now make sure that you understand the role of the stem cells in our small intestine. And then right now, how researchers were able to figure out that all of those cells in the intestinal lining are coming from the stem cells in the crypt. So this is a really clever experiment they did. They created a transgenic animals containing 2 transgenes. And I'm gonna go over this experiment because this is really nice, clever way of showing describing the method for tracking the cell lineage. So the first transgene is the GFP, driven by the stem cell specific promoter. So the stem cell specific promoter would be active only in the stem cells, and GFP would label the stem cells only. And also there is a gene for inactive recombinase, also driven by this stem cell specific promoter. So this is one transgene. And the second transgene is created by this reporter called LAC-Z, and this gene creates a blue pigment, but here they insert a blocking sequence here, and this gene is driven by the ubiquitous promoter. So ubiquitous promoter is active in every cell, but this blocking sequence prevents this blue dye formation. So what's happening when we have those 2 transgenes in the stem cell, this promoter will be active, will create GFP, so the stem cell, you can see stem cell glowing green, and also stem cell will produce this inactive recombinase. And when you provide the tamoxifen to the animal, the tamoxifen will activate the recombinase. Recombinase will come to this region, remove this blocking sequence, and by removing the blocking sequence, it will allow for the blue dye formation. So all of the cells derived from the stem cells will have ability to produce this blue dye. And that's exactly what they see in the experiments they did. So this is day 1, this is a cross-section, the immunofluorescent staining of the intestinal lining. Day 1, day 5, and day 60. And you can clearly see that all of those cells derived from the stem cells are producing blue dye. So it's like progressive movement of those cells upwards. And of course, we have stem cells in other tissues. We have stem cells in our muscles. And there are a variety of different muscles. We have hard muscle cells, we have smooth muscle cells, myoepithelial cells, and skeletal muscle fibers. And what we can see here, this is a skeletal muscle fiber shown here, and those green are the nuclei. So our skeletal muscles are multinucleated cells. And what you can see here on top, there is beautiful satellite cell. So this is the stem cell for skeletal muscle fibers. And this is a very important cell. And it's important for muscle regeneration. And what's happening? For example, when you go to the gym and you have a pretty intense workout, you can damage your muscle fibers. And this damage is this signal, sends the signal to those satellite cells, and they will become activated. So upon this damage, the satellite cells will activate and will start proliferating, producing those muscle progenitor cells. And the muscle progenitor cells can fuse back with the skeletal muscle fiber, and they can repair the damage, and the muscle can increase in size, so it can grow. When we do very intense workout, we damage our muscles and then activate the satellite cells, and they will help to repair the damage and build the muscle mass. That's how it happens. And of course, bone marrow is another source for different stem cells. Those are hematopoietic stem cells, and they are precursor of all of those blood cells and also cells of our immune system. So here we have a multipotent hematopoietic stem cell. It's located in the bone marrow, and it's the precursor for all of those lineages. And again, you don't need to know this graph or this diagram. You just have to know that we have a bone marrow stem cells that are precursors of the blood cells. But I do not require you to memorize this and know how hematopoietic stem cells, how they differentiate to those different cells. So do not worry about memorizing this. I won't require this information on the exam. But this is just to give you the example of another source of the stem cells in our body. And regarding the bone marrow stem cells, there was a very interesting experiment done in mice. And what the researchers did, they took a mouse and they X-ray irradiated the mouse. So they exposed the mouse to the X-ray irradiation, which completely blocked the blood cell production in this mouse. And this mouse would die within days, but they were able to rescue this animal by injecting a bone marrow cells from a healthy donor. So by injecting bone marrow cells from a healthy mouse, not exposed to the X-ray irradiation, this animal was saved by those bone marrow cells from a healthy donor, because they were able to replace the damaged cells and they were able to colonize the hematopoietic tissues and kind of regenerate the blood. And this is another very nice example, and it's in your textbook. It shows amazing abilities of stem cells for the regeneration of different organs. And in this case, this is the planarian worm, so very small, very simple worm living in the fresh water. And it has a very simple structure. It has 20 to 25 distinct differentiated cell types, so those are differentiated cells. And importantly, it has as much as 20% of the stem cells in this simple body. So they are called neoblasts, and they constitute 20% of all of those cells in this worm. And they have amazing regenerative potential. And what researchers did with this worm was quite amazing. So what they did, they took the worm, they exposed it to the X-ray irradiation, which pretty much killed all of the cells in the body. So they killed this worm. So here they have this dead body. And what they took a single cell from a healthy worm, they injected this single neoblast stem cell to this dead body, and over time, this cell was able to proliferate, differentiate, and pretty much restore a healthy, normal, healthy, functional organism. So regenerative potential in this animal is incredible. and quite amazing example. So all parts of the body could be regenerated from a single stem cell. And now I'm gonna move and talk about the induced pluripotent stem cells. So those are very important category of stem cells because this is another source of stem cells that we can create from our own adult cells, and we can use them in research, we can use them in the regenerative medicine. So there are a variety of different applications. But the good thing is that we don't have to worry about the destroying embryos. So this is an alternative source of stem cells. And they can be derived from the patient, from pretty much anybody, and they can grow we can culture them in the laboratory. We have the ability to differentiate them to different cells and use them in different applications. So and I will talk about the mechanisms of how do we go from the adult cell to the induced pluripotent stem cell in a little bit. But right now I wanted to show you this diagram. This is a very famous diagram when we talk about stem cells, when we talk about cell differentiation, cell reprogramming. We like to show this diagram, which shows like a landscape or like a hill and different valleys, and it represents all of those processes in a very nice way. So here what we have on top, we have a pluripotent stem cell, and this cell can differentiate into the different cells, right? So it can choose different paths. It can be differentiated to the muscle cell here, or to the nerve cell, or to different cells. So it pretty much goes from this unspecialized state here on top, and it can differentiate, acquire some specialized abilities, and it can choose only one path. So it's like a sequential reaction. It's like a so when this cell will determine the path, it cannot kind of go back. So this has to happen, the differentiation will happen. And here we have an example for a transdifferentiation. So we can have a terminally differentiated somatic cell that could be transdifferentiated to different cells. So for example, we can accomplish transdifferentiation of the liver cell to the neuronal cell. And this is of course in tightly controlled and regulated conditions in the laboratory. It doesn't happen naturally and spontaneously. It's a very controlled process. And of course, what regulates those the differentiation, we talk about the microenvironment of the landscape, and this could be the context of the tissue. So those cells, the stem cells, reside within specific tissues, and the tissue environment provides all of the clues and all of the important signals telling the cell to differentiate to the muscle or to the liver or to the different type of cell. So we talk about the microenvironment of the landscape, or microenvironment of the tissue, that determines what type of cell will be here or here. And of course we have this ability to go back, to go up the hill, to go from the terminally differentiated cell back to the pluripotent state. And this doesn't happen spontaneously, it doesn't happen in the adult body, but it can happen in laboratory. And researchers worked on that for a long time, for a very long time, and finally they were successful. Here, what are the mechanisms pulling the cells down the hill and causing cell differentiation? So I already talked about that. So in the tissue, in the context of tissue, we have physical and chemical signals, and also lots of transcription factors, growth factors. So all of those factors are key determinants telling the pluripotent cell to differentiate a specific state. And like I said, researchers were wondering, how can we go back? How can we go up the hill? Can we convert differentiated cell to a pluripotent embryonic stem cell-like state? And there was a huge breakthrough in science back in 2007, where a group of Japanese researchers presented their studies. They were able to induce pluripotent stem cells from the mouse embryonic and also mouse adult fibroblasts. And they discovered a key factors that played a major role in this conversion. And so this is the paper. They published their results in the Cell journal. It's a very prestigious journal. And yeah, that was back in 2007. And that was a major breakthrough in the science, opening very many doors to many different technologies. And right now, this whole research is exploding and it's expanding, and there is more and more discoveries, and we can generate we can generate stem cells from the adult cells, and we have pretty good understanding, or we can control the system, and the efficiency is increasing and improving. So this is how they were able to do it. So they took mouse embryonic fibroblasts, and what they knew that time, they knew that stem cells have specific genes that are expressed only in stem cells. So their idea was to put those genes on the viral vectors and transfect those genes to the adult cells and hope that perhaps those genes can do something, or that they can kind of reprogram adult cell to the stem cell state. So they were testing 24 genes that were critical embryonic stem cell genes. And when they were testing one by one, so they would put one gene to the adult cell, they were not successful. They were not successful. None of them was able to cause the conversion by itself. But when they started using combinations of different factors, they finally hit the jackpot. So they found that combination of 4 factors called OCT4, SOX2, KLF4, and MEC was necessary and sufficient for conversion of the mouse fibroblasts to pluripotent stem cells. And that time, that was the major breakthrough. This happened for the first time back in 2007. And those adult cells were able to convert to the stem cell, and those stem cells generated in this approach were very similar to the embryonic stem cells in very many aspects. So they behave pretty much like embryonic stem cells. So here we have those factors, OCT4, SOX2, KLF4, and MEC. And those are transcription factors. And like I said, we often refer to those factors as OSKM factors. And what they did in those, when they were overexpressed in the adult cell, they were able to act on the gene expression. So they were able to upregulate embryonic stem cell genes, and they were able to upregulate genes responsible for cell proliferation, and they were able to act on the chromatin structure. They were able to change the chromatin structure globally and loosen up the chromatin structure. And they were also able to downregulate the genes, the differentiation genes. So those 4 factors. And this is the outline of the experiment they did. So here we have a mouse fibroblast. And this mouse fibroblast was first transfected with the reporter gene. And this reporter gene is the G418. It carries the resistance for the geneticin. So this gene, when it's expressed, it allows cell to survive on the geneticin drug. And the geneticin blocks translation. So if cell cannot express this resistance gene, it will die. And this resistance gene was under the FBX15 promoter. This promoter is only active in the stem cells. So they created this fibroblast with this transgene, and they transfected those OSKM factors on the viral vectors. And then they plated those cells on the media containing geneticin. So all of those cells, adult cells, would die because they were not able to survive on the drug. And only the cells who were able to convert their state to the pluripotent stem cell-like, they were able to survive on the drug. And this process took quite a long time. It didn't happen overnight. It happened over the course of maybe a month or so, a couple of weeks. But they were able to find a couple of colonies that were surviving on the drug. And those cells, when they tested those cells, those were showing the typical features of the embryonic stem cells. So they were successful in their approach, but the efficiency of reprogramming was very low. It was less than 0.1%. And they, of course, performed a lot of tests on those induced pluripotent stem cells. They made sure that those new cells, they behaved like embryonic stem cells. And now, how much time we have? 15 minutes. Let's see. We can talk a little bit about about the reprogramming process, the mechanism of reprogramming, what is happening. So typically our adult cells are very resistant to reprogramming, and like I said, this process wouldn't happen spontaneously because cells, they have their own identity, and when we talk about cell identity, we mean that cells have a specific gene expression profile, which means that the neuronal cell is completely different. It has a different genes expressed when we compare it to the muscle cell, for example. And those different gene expression profiles are quite stable, so the cells can maintain their identity and specific functions. But cell identity can be changed, can be reprogrammed in vitro by forced changes to the transcriptional network. And this is the key in the reprogramming process, when we're trying to understand the mechanisms of reprogramming. And this is a really nice experiment showing how very early in the old days, how nuclei can be reprogrammed by transplantation into a foreign cytoplasm. So this is a very nice experiment done in frogs. When you have adult frog, you take skin cells, you grow them on the petri dish. So those are a terminally differentiated adult skin cells. And then you take a unfertilized egg, damage or remove the nucleus by exposure to the UV radiation. So the nucleus from the egg is lost here. And what they did, they took a somatic cell nucleus, they injected into this egg cell, and the nucleus from the somatic cell was reprogrammed by factors in the cytoplasm, and this would lead to normal embryo development and formation of normal organism. So the cell nucleus, even though it's in the differentiated cell, it contains a complete genome. So the information is there, and it's capable of supporting development of all normal cell types. And in this experiment, they demonstrated that oocyte cytoplasm can reprogram skin cell nucleus back to embryonic state. And of course, success rate was very low, because that time they didn't understand much about the reprogramming and what controls this process. But they were able to make a very important observations. So like I said, in a differentiated cells, they maintain their stable chromatin state and pattern of gene expression, and they cannot easily be changed. And reprogramming involves global changes in the chromatin structure. So during the reprogramming, the nucleus changes in shape and structure. It swells, it increases its volume 50-fold, and chromosomes decondense. And also there are a major alteration in patterns of DNA methylation and histone code. So all the modifications are erased and replaced with new modifications specific to the stem cells. So there is a global reset of the chromatin structure. And after all of those steps are accomplished, genome now becomes competent once again to initiate embryonic development and to give rise to differentiated cell types. Let's see. I think we're gonna continue with this the next lecture, but before that, we're gonna have a short quiz. So we're gonna distribute the index cards. So just you can just highlight one answer. So it's a multiple choice, very difficult question. It's not, do not worry. Done? So after you're done with the quiz, you can pick up your exam and talk with our TA.
Okay, so your exams, so that is, it's alphabetized, that's V through Z, last names, this is S and T, this is N through whatever letter is before S, Q, R, whatever, this is K through M, and then D through J, and A through D.
Yes, ma'am. Thank you.
I was pretty confused about one of the slides.
Once again, A through D,
The frog
E
experiment.
through
Does it like same as cloning?
J,
Like the
Cloning?
K
yeah,
through
about the same.
It's
N,
kind of similar.
Okay.
Yeah.
But it's still like a
through,
different process. It's called reprogramming.
R,
Yes,
S through T, and B
actually
through
this
Z.
experiment is described in the textbook. So during this lecture we talk about in the context of reprogramming.
Okay.
So yeah, but it's the same process pretty much.
Okay. Yeah, I'm just checking.
Yeah, okay.
Thank you.
You just need our name or ain't one of them.
Yes. Thank you.
Thank you.
All answers are correct.
I have a question. Can you walk me through this experiment again? The transgenic animals with the 2 transgenes.
Sure.
I'm sorry, it was just going
no,
really fast.
No. Yes, and actually this is in your textbook, so if for
Okay,
some
so,
reason
okay.
my explanation doesn't
Okay,
reach
it's in
you,
there, yeah.
Yes, but I'm happy. I'm happy.
Okay. After the craziness kind of dies down. How's your day going?
Yes, it's going.
It's
How's
good.
yours?
It's going, it's cold.
It's getting warmer.
It is gonna get warmer. I guess we needed a little bit of winter, you know.
Thank you.
But not in March.
Yeah, it has, yeah. Well, it could be like blackberry winter. We always get like the cold snap.
50, man. Proud of you. More than what I can say.
Did you get your exam? Not yet?
No. I don't want to look at it yet.
Do you feel you did good?
I
Better?
made above the average,
Oh,
so I'll
okay.
take that. But could have done better.
I don't know. Yeah, I'm seeing some of that. Okay.
Thank you.
Thank you.
Red cards? Thank you very much.
I think you're recording still.
Thank you.